Yes, we're recording. So with me is Dr. Joe Rhoades, joining here as for a talk about Aspergillosis. I'm Bart Kovart. I'm one of the trustees of the Aspergillosis Trust, and I obviously have ABPA myself. Dr. Rhoades, do you want to introduce yourself?
Thank you, Bart. Yes, thank you for having me. I am a genomic epidemiologist at Imperial College London. A genomic epidemiologist means I look at the DNA of pathogens to see how they are spread, how they are being transmitted, and also what drug resistance mutations they also have. So it's just a really fancy term for looking at the really fine scale parts of a pathogen and seeing what makes them tick. A lot of my research focuses on Aspergillus Fumigatus, which obviously is the causative agent of aspergillosis. I also do other work on other fungal pathogens as well.
Perfect. So you wrote this paper. Here it is. It's not light reading. But it's essentially about a drug-resistant aspergillosis infection by humans from the environment. So it is about this, how effective this is for Nox. Most people will know this. So let's just start with the beginning. You're the lead author. What did you want to prove with this paper, or what did you want to research?
So we have this idea that the use of fungicides, the dual use of fungicides in both agricultural settings, so in animal agriculture and also spraying on arable crops, but then the same sort of class of drugs being used clinically to treat people with aspergillosis isn't great, and that there essentially needs to be different drug classes used for different uses. And we wanted to prove that the use of agricultural fungicides is essentially causing Aspergillus to develop drug resistance mutations. And because Aspergillus sporulates, it goes into the air, that people are inhaling these drug resistant Aspergillus spores and then becoming ill. So that's what we wanted to prove here. We looked at just England, Wales, Scotland, and the Republic of Ireland for this study, and now we want to expand it worldwide.
So tell me about, so this, like, essentially you're saying fungicides are used in agriculture, are used for humans as well, and that has a negative impact on the resistance. So how did you sort of scientifically try to prove this?
So there are certain markers. So as I said, I'm a genomic epidemiologist. I look at the DNA sequence. So every single day, I'm looking at my screen. I'm looking at A's, T's, C's, and G's, the building blocks of every living thing on this planet. And I'm looking for certain parts that are essentially signatures of drug resistance. So we know that other people have done research that shows drug resistance due to certain fungicides. So we can search for those signatures in these data, in environmental samples that we've collected from farms, from soil, from parks. And then we can also look for them in clinical samples, so samples from patients. And what we tried to do was to essentially try to match the location. So for instance, we had a lot of samples from the south of Wales, from farms, from parks. We also tried to get clinical samples to match, same with London samples as well, because we wanted to show that people are picking these up locally, sort of like within a few miles radius. And that's what we showed. We could pick up these signatures in environmental samples, but also in patient.
Okay, so you create these data collections.
Yes.
And so if I understand this well, you try to have samples where there is intensive agriculture and likely to be fungicide used in agriculture, and comparing it to areas where you think that's less the case. Okay.
Yes, exactly. So it's intensive farming areas. So say for Wales, you grow a lot of potatoes there. So there's very intensive farming. But say for London, there's no farming, but there are open spaces. So there, for instance, it was like parks, Hyde Park, places like that. soil samples. So it wasn't necessarily farming, but there might be horticultural going on.
Oh yes, yes, yes. Okay.
We know that these drugs are used. It's the fungicides are used in horticulture massively as well. So it's different uses.
I see. Sorry, yeah, I didn't get that. Yeah, sorry.
And then it's important to note that this is a massive collaborative effort. It's the UK Public Health, it's the Welsh Public Health, Scottish Public Health, hospitals in Ireland. It's a massive collaborative effort to complete the study.
Yeah, you have an insane amount of data. I saw that. So when you have these populations, what did you see?
So what we saw was doing a really basic test called a principal component analysis. Sounds very fancy, but it's actually really simple. It looks at the genetics of populations that you want to compare, and we could see that the clinical samples and the environmental samples were very similar genetically, which means that they are essentially derived from the same population. They're the same population. So we can see that the Aspergillus that people are acquiring is the same as what we were picking up in the environment. So that was a very important thing to find.
Yeah. And what does that mean clinically? Like, because you're saying you have drug-resistant Aspergillus Fumigatus infection. What does that mean for patients?
So that means that if they're acquiring drug-resistant Aspergillus, then the drug treatments available become less.
Okay.
First line therapy of choice is itraconazole.
Yeah.
So if you're already acquiring a drug-resistant infection, then itraconazole might not actually be effective, and you might have to go to a different type of drug, which obviously, whilst doctors are figuring that out, costs you time, your things like that. And also other drugs, other drug classes are newer. They tend to be more expensive as well.
Yeah. So did you, and again, so I tried to read this, but it was a bit above my head. But did you, and I kind of gave up in the end, but did you also, so you saw the genomic correlation, but did you also see that in drug-resistant patients? Did you do that research as well, the prevalence?
Yeah, so not necessarily in this study, but we have done the drug-resistant prevalence environmentally and in patients, and that is in patients it's rising. It is rising environmentally, but we're starting to see an increase in drug resistance in patients in at-risk cohorts massively, especially cystic fibrosis patients.
Okay, so this research has proven the link. You as a scientist, you feel that the data is conclusive?
I feel that the data is conclusive, yes. As I said, we are looking to expand this now to see whether this is a UK only phenomenon. I don't think it is. So we're looking to expand this worldwide. So we've got a massive sampling effort going on worldwide now to get those clinical and environmental isolates. Yeah,
I actually, by the way, and this is not part of the study, but my understanding is that the Netherlands has a very big problem because they have a tulip growth.
Completely. Yeah, so anecdotally,
that's anecdotally, yes, yeah.
Tulip trade is megabucks, and the tulip bulbs get dunked in vats of azoles, fungicides, and shipped across the globe, of course. And tulip bulbs, they're a little bit like onions, aren't they? You can technically eat tulip bulbs, and the Aspergillus stays in the layers, and it can be exposed to these fungicides but not be killed, and that exposure is sufficient to allow it to develop drug resistance.
Okay.
But so the Netherlands has a problem, as do we, because we grow a lot of onions and we use a heck of a lot of azoles in our agriculture as well. Yeah, about 250,000 kilograms a year.
Yeah. So the study has proven that link, and so the next steps for you are to go international. And do you have any other areas that you're going to expand this?
So so go international. That's the next step. I want to prove the origin, not just spatially, so as in like location. So where did drug resistance arrive? When did it develop as well in terms of timing? Because if I can prove that it started like in the 1970s with when azoles were first used agriculturally, then that's kind of the final nail in the coffin for showing that. But we also know that there are drug resistance mutations present in Aspergillus that aren't these signatures that I'd expect to find, but the isolates are drug resistant. So there's something else. There's a different mechanism going on in some Aspergillus isolates, and it can evolve incredibly complex mutations. So that's also the next step, kind of to try and stay one step ahead, and hopefully that would feed into not just policy, but also developing new drugs.
Perfect. So I actually think, thank you very much for sort of summarizing this research. And I think for on behalf of our patients, I would say that this may look very dense, and it is, but it's actually really important. And I realize how much this is quoted and is like reviewed and spoken about. So, and you rightly say azoles are really important. They keep me well, like many other people. And if we will not have access to these, or if they will not be efficient, then that's going to be a big problem. So thank you very much for doing this research, and also thank you for taking the time to talk to us. We'll be keeping a close watch on what you will do next. Thank you very much.
Thank you.